Good evening to all the dignitaries and delegates. Thank you for joining us for today's webinar. My name is Dr. Devendra Kodati, and I am the convener and moderator for this webinar. St. Peter's Institute of Pharmacosciences is delighted to welcome you all for this international webinar on advances in drug discovery and development. This webinar is organized by Department of Pharmacology, St. Peter's Institute of Pharmacosciences in association with International Society for Pharmacoeconomics and Outcome Research ISPAR and Indian Pharmaceutical Association IPA Anmakonda local branch. We have received an overwhelming response from the participants, and I would like to thank everyone for displaying such great enthusiasm and participating in large number. We have crossed 1100 registrations for today's webinar from 66 pharmacy colleges across the globe. We have registrants from other countries like UK, US, Canada, and Malaysia. With this, I would now request Dr. P. Rashekar, Principal, St. Peter's, to deliver the welcome address. Okay, there are some technical issues. Now, without any further delay, we will start the scientific session. It's great pleasure for me to introduce our eminent speaker, Dr. Kumar Vyas Nemmani, Director, Sri Vishnu College Pharmacy, Bhimavaram, Andhra Pradesh. He has completed PhD from NIPER, Chandigarh in 2002 and completed PDF from McGill University, Montreal, Canada in 2005. Sir has published 40 plus peer-reviewed research articles and presented 20 plus national and international conferences. He is experienced pharmacologist with over 13 years of drug discovery research in pharma industries like Lupin, Piramel, Dr. Reddy's in different capacities. Sir has about 12 years academic research experience, expert at advancing novel discovery programs right from target selection to candidate nomination for clinical trials. Some of the nominated compounds are in advanced stages of clinical development. With this brief introduction, I would now welcome Sir to deliver a talk on pharmacological screening in drug discovery.
Yeah, so Devendra, can you see the presentation?
Yes, sir. Yes, sir.
Yeah, so my voice is clear?
Yes, sir. Yes, sir.
Yeah, so then, so first of all, thank you for contacting me and giving me this opportunity to present my favorite topic. So welcome all the participants for this webinar. I have taken this topic, pharmacological screening, drug discovery, and the reason is many of the pharmacists really dream of discovering new medicines for the treatment of various chronic disorders. Apart from this, this topic of drug discovery is present as a part of syllabus, name it whether it is brief form or in M form in any specialization and in PharmD courses. So given this as a topic of common across the courses and having my relevance experience, I thought of presenting this topic. So the time that is given is 30 minutes. So I will be rushing bit in giving the overview of drug discovery. And at the end, if you have any queries, you can ask me. So here, the topic that I'm presenting has been published by me recently as a book chapter um, in one of the reputed publishers. And the editor of this is Dr. Ramarao Poderi, is the ex-professor and the director of NIPER. So the topic I am presenting is majorly the concepts that has been described in this book chapter. Of course, there are a few additions and few animations and coloring to make the concept much more clearer to the participants. So if we start with the pharmacological screening drug discovery, let me start with describing what is pharmacology and what is discovery. And then we'll get into details step by step what's all done as part of the screening. So as you all know, the pharmacology is nothing but pharmacokinetics and dynamics. In other words, we define it as what drug does to the body as pharmacodynamics and what body does to the drug as pharmacokinetics. So when the drug is given to the subject, it gets absorbed and get distributed and undergoes metabolism, of course, elimination. During this process, when it reaches the site of action, it starts eliciting its dynamic action. This dynamic action could be leading to a primary pharmacology, what we call it as a desired response, which is called as activity. And it also may lead to some side effects. We call it as a undesired effect or side effect or unwanted effect. And it could be due to the primary target mechanism of action, or it could be due to off-target activity of the compound. So in the discovery, though we are very much interested in developing or understanding the pharmacology of the compound, and the other side of the story, or the other side of the coin, is toxicology. So the same principles applies for the toxicology as well. The drug needs to get absorbed, needs to be distributed, and needs to reach the site of action and produce its effect. So the famous quote that comes to the mind when thinks of the toxicity studies is, the dose makes the poison. So all that the magic is the difference in pharmacology and the toxicology is the dose. So the right dose, you get elicit the pharmacological activity. When the dose reaches a certain limit, it might lead to the side effects of the toxicity. The principles are the same, right? So with this, let's see what is pharmacokinetics. The ultimate goal in a discovery program is to find the right drug, of course, at the right dose, and we have to give it at the right time to the right patient. So if a drug is given to a wrong patient, it might lead to some complications. Of course, it all depends on the kinetic principles of the compound. The wrong time, you may not achieve activity. And wrong dose, yes, it might lead to side effects, toxicity, or it might not show activity. So the objective in making this pharmacological screening is to find the right drug first, and find the right time, and find the right dose, and to be given to the right patients. Keeping these four objectives, the studies are planned and designed and rationalized in doing those studies. So thorough understanding of this pharmacokinetic profile will not only minimize the risk of drug attrition, but also aid in optimal use. That's the reason we'll be very much focused in understanding or studying the pharmacokinetics of the compound. So if a pharmacokinetics is not right, the molecule may be withdrawn at a later stage, or it may not produce its optimal activity or optimal potential, you may not be able to use it. So this is the main goal in doing the kinetics. Let's see the dynamics part, pharmacodynamics. So in a discovery point of view, the dynamics is we have to make a molecule which meet an unmet medical need. So unmet medical need, let me make it clear, for analgesics, we all know they all produces relief pain, but if you ask the patients, every 2 in 3 patients say they're not satisfactory with the drug treatment for their chronic pain. And they're not satisfied because it's not producing enough pharmacological activity. They're not satisfied because it is producing unwanted side effects like, so if you take the NSAIDs, we get gastric irritation, increase in BP, and cardiovascular complications. That is something which comes as a baggage with the pharmacological activity. So here, there is definitely need for good analgesics, which is either very effective or at least should not have those side effects which are there with currently existing drugs. That is the unmet medical need. And another important thing is in the discovery, when the scientists dream of about a molecule, it has to be safe and very effective. And at the same time, it should be easily administrable. Cost, of course, is another aspect. If the profile is so good, cost is not a bar for the success of the molecule from industry point of view in the market. So the dynamic studies, when you talk about it, there are two terms that is given analogy. The right molecule is the key, and the right target is the lock. So you should have a lock, and you should find a key by doing trial and error and able to open up the key, that lock. So finding the lock is the most critical part in this business. So if you see how drugs are discovered in the past and the present system is called more of modern drug discovery, and the past is more of a traditional drug discovery. The past, I'm talking about 30 years or 50 years back. And those days, the drugs are discovered by phenotypic-based screening. Phenotypic-based screening means the drugs are directly screened on the human beings or the living systems. And then whether it is working or not. If it is working, and later with time, people to understand how it is working and what are its mechanisms of action and so on. But the present modern drug discovery is not going in that direction. Here we start with the right lock, the right target, and then start making molecules which will able to open the lock effectively and in a desired way. And we receive the decent pharmacological activity with minimal toxicity. And if you see the present case of Anandaya, that medication for COVID, that's more like a traditional system. It's an Ayurvedic, it's a traditional. The medication is made and it is directly given to the subjects. And it's truly so, many people worried more about whether it is working or not, but more about whether it is safe enough or not. We all know that COVID, even without treatment, it goes away. At least 95% of the subjects, but 5% definitely need a proper care. But if drug is having a side effect, then it might lead to 100% of the subjects' complications. So that is the reason that phenotypic-based screening is not a right approach as per the current application of the science is concerned. Of course, there are few advantages with that, but disadvantages are much more. Let's understand more about modern drug discovery. So what the past is telling us, if you do the traditional screening, that's more like finding a needle from a haystack, and whereas modern screening is more like a target. You specifically aim the target and keep on trying and meet that goal. So traditional has given the first-in-class compounds, that is first of its kind. If you see the examples, I can name it like metformin for diabetes. So there's no second metformin. So people are not able to do it, and people doesn't know what the actual target is even today. But of course, we have some pharmacology understanding, but the molecular target or the major target molecule, we don't know it. It has been discovered through traditional. And same is the case with the NSAIDs. It's all started with natural products box and then trying and error. It is working, and later it was found to be salicylate is part of that, and then it has come aspirin. So whereas in modern discovery, it is best-in-class compounds. You have a compound, say the innovator has come with the compound, and you can make the much more rationalized screening and find or tweak the efficacy and the safety parameters of that compound and generate more Me Too drugs. And such a strategy is very much easier with the current approach of modern drug discovery. So now we understood what is traditional modern, and we are in the modern world. So let's understand more about the modern discovery. In that, very first and most important, the difficult challenge is selecting the right target, the right lock. As per a study, and we have about 19,613 proteins. Out of that, 14,545 are non-disease causing proteins. It means these proteins are not altered in disease condition. Rather, this disease is not caused by these proteins. And there are few 3,000 odd targets or proteins which cannot be manipulated or modulated with the drugs. So what has been so far successful, according to the report, that is a case analysis, Human Protein Atlas, you can see that website, is only 3%, that 670 proteins have been successfully addressed so far, and it's only 3%. And there is about 7% is still not addressed. There is 1,265 proteins are there. So when there are 1,000 proteins, it is like finding the sky, looking at the sky and finding which star is the best star to look at and to aim at. So 1,000 proteins are there, and when start looking at the specific protein in depth, and obvious that we'll be missing some new proteins or new understanding in the literature. So finding that right target, that there's a process called pre-discovery, which we are not discussing in this talk because it's a very complicated and very interesting story. Maybe some other time we'll discuss that. So once you select the target, then what our steps needs to be done is what we'll discuss. So this is what we'll see. The new drug discovery has mainly these stages. First is called high throughput screening. And in this process, here we test all the library of compounds. The libraries are made in the organizations, like anything and every compound that is made, and the intermediates that's made are being stored in the organization, and they make it as a library. And those libraries are used against the selected target. And since these libraries are in hundreds of, maybe in thousands, and in multinational companies, they're in millions, so we need to find which one is the one which is likely to hit the target, even at low concentration is fine. It is only possible by using high throughput screening. We'll see what is high throughput screening, how people use this robotic technology in screening millions of compounds in shorter period, like in few months, right? So that process we do, and we get the hit, and we call them those are hits. Then we understanding the hit, and we're selecting the few hits which are having a decent potency, and start looking at the any structure-act relationship is there among the hits, and is there any patent scope. By the chemists, we do all that analysis, and they select few compounds or few scaffolds, and that they generate, apart from this potency, some of the assays that's mentioned here, like in vitro secondary assays, and then look at the ADME properties, and then look at the preliminary pharmacokinetics, and even basic toxicology studies. That stage we call it as a lead. And these leads, once you generate, and again, this is a filtration process, and it is a reiteration process, and we select few leads. And these few leads will be selected based on the strengths and the weaknesses of that organization in manipulating that molecule. What they do is very focused chemistry, taking that molecule, making the analogs, and then testing further in these assays. Here you note that the symbol here, then you do the lead optimization. If none of them is not meeting the criteria, then you might go back into the lead stage and find novel scaffolds. This is an iteration process. So once you made that lead optimization, and what is lead optimization, those criteria we'll see at the end. Once you're satisfied with that, then you take the few compounds as a preclinical candidates. At this stage, we start seeing or analyzing the GLP toxicology. Now we know at the stage of lead optimization, the compound is having desired potency, desired selectivity, optimal PK parameters, and decent efficacy in animal models. Now in this preclinical candidate stage, we'll undergo this selected compounds into a GLP toxicology studies, and we do extensive pharmacokinetic studies like interspecies scaling studies to find the first time in human dose calculations, and then extensive toxicokinetic studies to find the relationship between the concentration and the toxicity that is elicited by the compound. So after a few compounds, maybe one or two compounds, you select for the clinical development. That means select the compound and submitting for an IND. So in this case, you make the compound in large quantities, you write a clinical trial protocol, and make an IB report having all the summaries of this, and submit to the regulatory authority for approval. That is the beginning of a new story. That is the drug development. So we limit to drug discovery at this stage. These are the various steps, and these are various milestones, and these are the various assays and the protocols that is done as a part of this big game. So now let's see high throughput screening. Okay, so now we said that high throughput screening is done. So it is done using this lab air, and it is mostly done in MTPs, microtiter plates. They are usually 96 and multiples of that 96. There's been 386 and the multiples of that. So usually screening is done at one concentration at high dose and in duplicates. So in 96 plate, we can screen 48 compounds. So if in 386, so multiples of 4. All that is done at one time pipetting using this robotic arm. So this is a compound storage station. You just keep the compounds and everything. And this is a handling station, and this is a complete robotic system where you just have to start remotely, not even go inside. When keep everything arranged and remotely by just clickings, that robotic arms takes the plate out and do the all the handling procedures, liquid handling procedures, and finally put it in the plate reader, and the readout is come out and it is recorded in the computer. That's how these are all high robotic arms are available. And this right now what I'm showing is a very simplistic automation, but high-end and continuous processing robotic systems are used in multinational organizations to screen 2 million, 3 million compounds in a matter of 1 month or 2 months or so. That's it. All compounds are screened and they have an idea. They select few compounds from that exercise. So this is basically doing this by faster manner, identifying the hits. So then once you find the hit, and you need to confirm it by doing manually, and these assays called in vitro assays. Usually the primary assay we call it, which is a cell-free system. It means directly taking the protein and see your compound is inhibiting the activity of that protein. So give a substrate and see the product given enzyme, which is the protein in this case. And if a compound is a inhibitor of that enzyme, whether you see that substrate is conversion to the product is being inhibited or not. That is simple homogeneous assays are being used. These assays are used in high throughput platform and can be done and also done manually to confirm it, and also done at multiple concentration to confirm the hits what you see on the machine, on the robotic system is true or not using this primary assays. The advantage is here, this is a target. We are working, we know the target, and to that target we are seeing miracle is altering that target or not. Then is the secondary assays. These are cell-based assays. Means you're taking whole cell system. So this is more of biologically relevant compared to this primary assay, but it's complicated because it's a cell system and you have to lyse the cell at the end. If it is a changes happens in the cytoplasm, if it is a secreting protein, then you can estimate in the media what is that protein is. All this analysis using high-end analytical techniques. So here is the analysis people important. Here is the molecular biologists or pharmacologists are required. Using these technologies, developing the assays and quantifying it. And these are various techniques and technologies which will not go depth as of now in the interest of the time limit. So once the HTS is done, the primary in vitro and second in vitro assays are being to confirm that potency and selectivity and specificity of the compound. So then comes as the ADME and the DMPK. Absorption, distribution, metabolism, called Tier 1, Type 2, and Tier 1, Type 2. In ADME studies, is basically look at whether a compound is stable in biological fluid, whether a compound has protein binding property. Most of the drugs do bind, it does bind to the albumin, and basic does bind to the keto acids. And we see that whether compound is being metabolized by the microsomal enzymes. In this case, we take microsomes from different species and to see which species kinetics are similar to the human metabolism. Means you take human liver microsomes are commercially available, and you also test the different animals tissues and then see whether metabolism is taking place or not, and which mouse, which species data is matching with human. That species is used for selecting the further studies, whether it is a kinetic study, whether it is a dynamic study, whether it is toxicology studies. That's how doing this in vitro ADME studies is very critical for selecting the species and extrapolating the data to the human. Once you understand this stability and metabolism, and we look at whether compound is an enzyme inhibitor or an enzyme inducer or as a substrate for an enzyme. So this data will help us in understanding whether it is a protein-protein drug interaction is possible or not. Followed by that, we do a typical kinetic study and we estimate the various parameters. At the same time, we like to see the tissue distribution, how this compound or which tissues the compound is moving in. Once data is so good in all these parameters, then we go for a very decent or comprehensive pharmacokinetic studies. We call single ascending dose and multiple ascending dose, means increasing the doses in different animals and see whether the saturation kinetics is taking place or not. And repeat dose studies we do and to compare the day 1 kinetics versus day 30 to see whether there is an enzyme induction and enzyme inhibition is taking place or not, based on the comparing the area under the curves of the compound. Then we also do some mechanistic studies, whether the drug is being disposed of from the bile or it is undergoes first pass metabolism using this portal vein cannulated studies. These are some mechanistic studies we do as a part of this comprehensive pharmacokinetics. So basically, these studies will help us in selecting this right species, understanding or predicting the drug-drug interaction possible, and more importantly, in estimating the first time in human dose, basing on this um, um, ADME and DMPK studies. Because giving the dose first into the human as a part of the development is a very critical step, and it has to be very safe, and then it has to be effective. So regulators are more worried of the safety. So all the safety estimations and projections of the first dose is basing on the data generated as a part of this package. So comes to the species, and you see, I love this slide. Various species, and each species has its own advantages and disadvantages, and some are more suitable for something, not for others. So if we take the pig, and the pig has a heart similar to human body, so the ideal species for doing the cardiac activities is pig is the most suitable, and guinea pig is very sensitive to the and very similar to the human. So that's a good model for the respiratory compounds. And hamsters are very good in terms of the um, metabolism of the cholesterol. So if atherosclerosis or anything you are interested in, hamsters are the suitable species. That way, and monkeys are the best for they are like humans. They have that emotions, they have empathy, and CNS drugs are best screened and tested in the monkeys. And which species you select, basing on all the TMPK study that you have done, basing on that you select the species. Not only species selection, very important is if you identify that the rat as a suitable or mimicking the human kinetics and in terms of the major active ingredient in terms of the metabolites, and within the rats also there are various strains and which are very specific for a particular disease. If you are interested in diabetes and if it is an insulin sensitive compound, the GK rats are the ideal compounds, or Zucker diabetic fatty rats. They show exact human pathophysiology. What we see in humans is what happens in these ones. These are genetically um, spontaneous mutations, and from birth to the adult and mature, the stages that we see in humans, development of type 2 diabetes is exactly mimics in these rats, and they are the most suitable rat strains for testing this screening these anti-diabetic compounds. That way, every disease has there's a good amount of knowledge which strain or which species is ideal for a given mechanism of action. Very important is this rat strains is critical for positioning your compound in the market. How you share that what are the existing compounds, how your compound is different from the existing compounds in terms of the mechanism, in terms of the activity. These models help in doing that. So this needs an expertise. This is what I've done in my industry. I'm very happy in discussing much more about this. If anybody have any question on that. So then these are the mouse strains. You see here, this is a diabetic mouse. This is a obese mouse. By looking at that phenotype, you can decide whether it is obese or not. And for obesity screening, these mice are the ideal ones. Of course, you need to keep in mind the target and the target pathway and the target outcome. Basing on that, you need to select the right species and the right strain for a given study, right? So having seen that test, now with this so far what we have understood, we screen the compound in stage to get the good potency, and we screen the compound in vitro primary assay and secondary assay to see that the potency is confirmed, and we see that compound is selective, so compound is specific, and we see that compound has desirable PK properties, and since all and having desired activity in animal models. So once you see that all the package is so good, in such a case, then you start exploring the safety of the compound. And basically, this is a core battery, CNS, cardiovascular, respiratory. These are basic pharmacology studies, and pharmacologists usually do industry these studies. And some additional organs, also like GI and liver, and those studies also one can do more as a part of the safety pharmacology studies. So far we ensured that activity, and then the kinetics, and then safety of the compound using this battery of tests. Then comes is if all these properties are so good, we call it as a lead. Now we have to make lead optimization. In this case, we want to make the compound even much more better. If there is any weakness in any one of the parameter, this is the time point where these optimizations are done. So it's a very tough challenge, lead optimization, because here there are 6 various parameters which we are talking, and all 6 have to maintain. When start modulating a compound, when you're making analogs, maybe one is fixed but one goes off. But here, a delicate balance of all 6 parameters needs to be made. This is a very challenging process. Medicinal chemists needs to do a great job in association and working with the biologists. Here, we want to fix all the problems. Basing on that, we select the compounds, and then we go for the early studies. In this lead optimization stage, what the famous quote is, fail cheap and fail early. If you're not happy with the compound, don't develop it. But if you compromise and you develop it, at later stages, you go on ending spending a lot of money, and you find it is not good enough, then you end up with losing time and money, which should not happen. Right? Basing on that fail cheap, fail early is you have to follow that lead optimization. You have to be patient and do all the rigorous exercises and see that all parameters are fixed. So now let's see this each stage. What is the criteria? Numbers. So in hit stage, we want to see that the compound is of known purity and it is having a decent potency. So once you do the more characterization, especially preliminary ADME studies, and you see that the kinetics also decent enough and it is selective and specific. Apart from that, chemist looks at is whether there is a tracking of the SAR, a structure-activity relationship. By modulating some structural features, are you maintaining the activity? Are you getting better activity? Are you getting better safety? Are you getting better absorption? Whatever that is that you're interested in, are you able to track it? If there's no tracking, it becomes like lottery. Like no way next thing you can predict. So that is the reason chemist looks at SAR profile and select those having SAR profile only, not the compound that meeting all the criteria. Very, very important property. So then is the lead optimization. In this case, as we discussed, apart from the preliminary activity, safety, kinetics, we also look at the efficacy of the compound. We also look at the safety of the compound. We also look at the toxicity of the compound. If all are fixed, then we do some more characterization and do the large scale and proper GLP toxicology. This is the stage we met the criteria. We should have a compound with low nanomolar IC50, low nanomolar, and then it should be highly specific and selective. The IC50 is more than 20 times. Of course, it varies from target to target, and it should not inhibit any of the cytochrome P450s, IC50s, at least tenfold compared to the IC50s in efficacy. And even at 10 micromolar, it should not any enzyme induction or enzyme inhibition properties. And you should not be inhibit any of the enzymes even at 20 micromolars. And you should have optical parameters. You should have oral belt more than 20%. And these are all like ideal castics. It is difficult to get all good castics in one molecule, but that is where that challenge comes in the thing. So once you have all this data and you have seen the pharmacology and toxicology and the CMC and the clinical trial protocol, based on data, you write the clinical trial protocol, and then you write a clinical trial protocol and write the IB report and submit as an IND. This completes the discovery part. And this is the beginning of a new story, that is clinical development. So with this exactly in 30 minutes, I completed my presentation in the time slot that is given. I'd like to thank you all for your time and giving this opportunity. So for doing all this kind of work, you need a good facility like this. This is the facility we set up at Vishnu College of Pharmacy in Bhimavaram. And this is the front view, and this is the breeding area, this is the dirty corridor, this is the clean corridor. We have mice room, sterilization, necropsy, and we have the rats room, wash area. We're all just setting it up, this facility. We're almost done. Due to COVID, it got a bit delayed than the set timelines. This facility is up running, and you see the facility. We call this as Center for Preclinical Research Excellence. If any of you are really interested in doing your work, if you can bear the cost, we welcome you to come and work in this facility. And if you want to do as a collaborative work, we welcome that model as well. And here is the coordinator, Dr. Pawan Kumar Samudrala. Here is the mobile number and the email ID. If anyone are interested in doing this pharmacology work, what we have discussed is being wanting to practice and implement in this facility. So this is what I want to present in this. And thank you all for your time. I'm happy to take any questions from your end. So over to Dr. Devender.
Yeah, thank you, sir. I hope all enjoyed this amazing presentation. It was pleasure to have you with us, sir. If any
Yeah,
questions, I'll get back to you, sir.
yeah, thank you.
Thank you once again, sir. Now I would now request our chairman, Sri T. Jayapalan sir, to enlighten us with his presidential address.
Very good evening to you all, and I'm so delighted listening the entire session of Dr. Kumar from Vishnu College. I'm so attached to Vishnu College, and it was a very sequential kind of wonderful presentation. Your lecture is so tempting and very enlightening. Really, I thank you for sparing your time and enlightening all the delegates in this particular web conference. And it was very clear and fantastic. Even I'm not from pharmacy, but still I'm able to learn a lot from your session. Thank you, sir, for a very, very fantastic presentation on these particular advances in drug discovery and development. Because these basic things are very much important for the youngsters who are going to come into the drug discovery research. And uh, all my good wishes to entire Bhimvaram team. Even I visited your college a couple of years back. I know everyone and your intensity of your knowledge, and the people around in the Vishnu campus, and I'm so delighted with your presence in this opening session of this particular conference. Thank you, sir, once again from my side. And the beauty of this wonderful session, I think I should really appreciate Dr. Devender, who is the convener and head of the Department of Pharmacology, for taking initiative and organizing this particular conference, and also the chair, Dr. Rajsekhar, who is the principal of St.Peter's Institute of Pharmaceutical Sciences, and of course the entire team of organizing, Mr. Santosh and Anusha, who are also taking care of this particular conference, and the coming speakers who are also going to talk on human genetics and drug discovery by Dr. Gautam Matla, who is also from GSK London, and followed by bioequivalent studies in drug development by Raghavendra, who is from Strides. So I think all your experiences, it really counts for all the youngsters and all the delegates who are coming from the college across the country and also from other parts of the world. I think that this session, this particular web conference, I think it's going to enlighten, give us a ray of hope. They're going to have the clarity of what is going to start from where and what are the things they have to take into consideration while getting into the drug discovery research and development. I think India today, already I told in the previous conference, world is looking at India now. Okay, and definitely Dr. Kumar must be knowing when you are working in Canada, day by day, year by year, Indians are getting a lot of huge respect because of their scientific temperament. Even the government of India is pushing it a lot. They want to see because at this particular scenario of this COVID, India is the only country which has done in the fast track to come up with the vaccines. Not only just vaccines, even saving lives, the drugs to get out of the COVID. So we are in the front now. I think you people, I think all pharmacists, all the professors, all the research scientists, and all the industries, we all as a team, I think we made a huge impact across the globe, not just India. So I really salute all the people who are really making their contribution somewhere or other. Teachers must be contributing uh, for all the young people to come into the next level of their research, bringing some ray of hope to different kind of diseases, different kind of viruses across the globe. Dear students, I think when I see the global opportunities, I think now every country they want to have at least one or two research scientists in their team in their countries. So that is the impact right now. The last 1 or 2 years, the country has given. Country has given a message. So this is a big opportunity for you all. Not just India alone. You are being welcomed. No, many now Indians are being taken in PDA, postdoctoral fellowship programs. Many Indians, even MPhones, are being welcomed to do their research in their countries because they believe in our knowledge, they believe in our scientific improvement, they believe in your what you call the kind of learning that is happening in this country. So with this short message, I think I want to really appreciate your involvement, your learning, and because tomorrow the future is yours. Because of you, I think world is going to be really saved, and definitely India is giving at the lowest possible what you call price, low priced medicines and 100% efficacy. Okay, and even today, I think yesterday you might have seen even our Prime Minister is so positive, where he want to see that Indians has to contribute in the global Indian economy and in the exports. I think right now, pharma, we are touching close to 13% of the exports in the Indian total business economy. I think in one decade, less than a decade, I think we are going to touch into, I don't know, might be crossing 16, 17 or 18%. I think you people are contributing to this India's economy, India's growth, India's stability, and even you people can make an impact to bring the prices of this country down. You are going to make this thing stable. You are going to make healthier India. Okay, that's very much important more than money. Okay, so I really once again I want all the delegates please use this platform. Every speaker who is coming here, I think they have done their huge research. There's a lot of broader knowledge. They are here to help us to learn to go to the next level. Okay, so I wish you once again for a better learning and this to have a one more successful web conference. Take advantage of this, and I really thank all the speakers for accepting and to deliver the lecture for all of the delegates. Thank you once again. Thank you, sir.
So thank you, sir. Thank you for your nice words. I've seen your contribution as president of the various Indian organizations. You are so active and initiating and taking these activities, and we'll be very happy to be part of it. And thanks again for giving me this opportunity. And,
Thank you. Thanks
very
for
nice
time.
message. Very nice message for all the participants. Look
Thank you, sir.
forward to
Thank
interact to meet you sometime soon. Yeah.
Sure, sir. Definitely. I look forward. Thank you.
Thank you, sir. Now let's move on to our second question. Now let's move on to our second session. I would request Mrs. P. Anusha, Assistant Professor, St. Peter's Institute of Pharmacy Sciences, to introduce our next speaker, Dr. Gautam Atla.
A very good evening to all and welcome to today's session 2. It's great pleasure for me to introduce our speaker today, who is going to talk about human genetics and drug discovery, Dr. Gautam Atla. Dr. Gautam Atla did his master's in pharmacoinformatics, NIPER, Kolkata. Later, he spent a couple of years in National Center for Biological Sciences in Bangalore as computational biologist before moving to Europe. He received prestigious Marie Curie Fellowship to pursue his PhD. He did his PhD in Center for Genomic Regulation in Barcelona, Spain, in collaboration with Imperial College London under the supervision of Professor George Ferrer. His PhD was mainly focused on understanding genetic regulatory mechanisms of type 2 diabetes. Now, he is working as a research scientist in drug discovery, R&D, human genetics team in the GlaxoSmithKline, London. I request her to start the session.
Can you see my screen?
Yes, sir.
Okay, cool. So I'm going to make it full screen. Do you see full screen?
Yes, sir, it is visible, sir.
Thanks. There is some background noise, but maybe if everybody mutes, that would be very useful.
It's coming.
Yeah, cool. So so there is some background noise. If everybody mutes themselves, that will be useful, because I hear like some noise.
No, it's
Okay,
clear,
so
sir.
okay, cool. So I'm going to start talking about. So the topic that I have given is human genetics and drug discovery. So, but what I will do in this session is to give a very high-level overview of what is the thing is about human genomics and human genetics and how it is being used in healthcare. Because recently there is a lot of emphasis from various pharmaceutical companies to invest in human genetics studies to advance drug discovery research. So before going directly into human genetics and drug discovery, I will try to explain a little bit of what is human genomics, what is human genetics, and because I'm not sure if the audience or the students are very familiar with all these concepts, because they are very recent and it's not easy to get head around them. So I'll give some introduction and give some very basic overview, and maybe later, if you have any questions, you can contact me regarding these topics. So I would like to start with to ask a question with why, if you look at any two individuals or any individual, they're all different phenotypically. Phenotypically in the sense they have different hair color, different eye color, the skin color is different, some are tall, some are short. So every individual is very different, right? And even not only at individual level, but if you also look at cell types, you have a lot of cell types in your body, and each cell type has a different function. So why each cell knows what it has to do, or why there are different cell types and who is instructing them to perform specific functions, right? So in order to understand these things, first I'll explain a little bit about what is human genome. The human genome means each and every living organism, not only humans, have this thing called nucleic acid, which is called DNA or RNA. So DNA, each cell has a DNA that is, in humans, it's around 3 billion nucleotides long. So it's composed of nucleotides, which are called base pairs, and it is 3 billion nucleotide longs that is wrapped around nucleosomes, and then it's further compacted into chromosomes. So the importance of human DNA is that the central dogma of molecular biology tells us that the sequence of the DNA is transcribed to mRNA, and the mRNA is ultimately translated to proteins. So depending on the sequence of the DNA where this protein is coming from, we have different proteins with different amino acid sequences and different functions. It's been 20 years that the first human genome project has been completed, which revealed that there are 3 billion nucleotides in our human genome, and there are 23 pairs of chromosomes. And these 3 billion nucleotides, or the genome or the DNA sequence of humans, encodes for more than 20,000 genes, and that gives rise to more than 100,000 proteins. And there are over 1 million regulators that regulate this protein expression or gene expression in the human genome. So going back to the question why each cell performs different functions, or why each cell is different, because even though each and every cell in the human body has the same DNA sequence, the sequence of the DNA is exactly similar across all these cells, but they have different functions. Because the parts of the DNA, let's imagine this is a DNA sequence and this is a gene. The parts of the DNA that are active in different cells is different. For example, if it is immune cells, there are genes that are relevant for immune functions such as cytokines production are active. In the microglia, maybe the regions of the genome that encodes for dopaminergic receptors are active. In case of, for example, pancreas, the genes, the parts of the genome where insulin is produced or insulin gene is transcribed is active. So because all of them, even though all of them has same DNA sequence, which regions of the DNA sequence are active in which cell types determines what's their function. But we have hundreds of tissues and thousands of cell types in human body, and each, again, the DNA is the same in each cell type, but they have like different functions. So there is a whole field called regulatory genomics that try to understand how this complexity of cell type function is established in human body, right? And how precisely each and every cell knows, how the pancreas knows it has to secrete insulin, why brain cells knows they have to secrete dopamine, why not the brain cells secrete insulin or pancreas secrete dopamine, right? So they know precisely what is their function, when they have to active, when they should be active, and when they should secrete or when should they perform their function. So all this complexity of regulation of the cell function is studied under like huge umbrella of subject called regulatory genomics, and this is like, it's by itself is a massive field, and I'm not going to explain much about what is regulatory genome. But one thing is, I want to mention that the human genome sequence is 3 billion nucleotide long. So only 22% of the human genome encodes for proteins, and the rest of the genome has these something called regulatory sequences or regulators that determines which genes and when they should be active in which cell type. So that, for example, they're called enhancers or regulatory regions, or there are different terms. So that's how complex, if you start looking at the DNA level, that's how complex it gets, the genome architecture. So going back to the question, why each person is phenotypically different? Because if you look at the DNA sequence of any two people, there are 4 million differences in the DNA sequence. That means 4 million is like 40 lakhs. So there are 40 lakh nucleotide differences between any two people you take in the world. There are at least 4 million nucleotide differences. Because of the differences in this DNA sequence between two persons, there is possibility that each person has a different characteristics phenotypically, like how they look like, how their hair color look like, how their eye color looks like, whether they should be tall or whether they should be short, whether they should be dark skinned or light skinned. So everything is encoded in this genome, in this DNA, and differences in the DNA sequences gives rise to different combinations of phenotypes. So not only that, because of these DNA sequence differences, which we call as genetic variation or SNPs, the terms are used as genetic variation or DNA sequence differences, not only gives rise to physical differences like eye color or height, but because of the difference in the genomic sequences in each and every individual, that also makes them either more or less prone to several diseases. So depending on the genetic background, it's possible that some people might be at more risk of getting a heart or diabetes or immune-related disease or neuro-related diseases, or some people are less prone to get diabetes. For example, if you see, not everybody has type 2 diabetes or not everybody has a heart attack, right? Like only some people get, some people don't get. Okay, so certain aspect of this could be environmental, like what food they eat, how active they are. There is some certain component of environment, but there is a large component of genetics as well. So genetics also determines if a person gets a diabetes or if a person is more risk of getting cardiovascular disease. So why the genetics plays a role in disease susceptibility? I'll illustrate with one very basic example is that this is let's imagine this is a genome sequence, this is a DNA, and there's a region where the insulin gene is produced. So if everything is perfect, you get insulin and normal insulin. So when you eat food or some cookies or bread, your glucose levels goes up, and this is normal DNA. So the pancreas produces insulin and your blood glucose levels are controlled. Imagine that there is a person that has a mutation or the change in the DNA sequence where the insulin gene is produced. In that person, either they produce less insulin or they produce a defective insulin gene because there is a change or there is a mutation in the genome sequence of that person. And those people get various types of diabetes, and they cannot, when they eat food or when they eat like cookies or chocolates, their blood level will not be controlled normally because the insulin gene is not normally expressed or not normal levels because they have mutations. So they take insulin or other medication that will increase the insulin sensitivity or increase the insulin production, but they get diabetes at the end. So that's how the genetic mutations or the genetic variation between individuals contributes to different diseases. So there is a genetic component whether people get a disease or not. But this is very how do I say, like very, very simple example that I'm showing. And if you just think about insulin, the release of insulin and glucose sensing is a whole complex pathway. So when the cell senses blood glucose level until it secretes insulin, there is a lot of components and moving parts and the pathways that operate. So the genetic variation, not only insulin, but if the genetic variation causes any problems or effects in any of these genes that encodes for these enzymes, that proteins, etc., that person will have or will develop or is at high risk of getting diabetes. So it's not just about insulin, it's about all the pathways that are involved in glucose sensing to insulin secretion. Any defects in the DNA sequences that encode for any of these proteins or genes will cause diabetes at the end. So that's how complex it is, and it is just one example of insulin diabetes, but you have like thousands of diseases that are caused because of the dysfunction of the cell, and that is the underlying cause is DNA sequence changes. So how do you understand how this genetic variation or the changes in the DNA causes disease or not? Because not all changes are important, because the changes occurs in a very important gene or important protein that causes disease. So first you need to understand which of the genetic changes causes which diseases, right? Because as we saw before, certain parts of the genome encodes for insulin and certain parts of the genome are active in brain. So if this change occurs in insulin producing parts of the DNA, you get diabetes. But if this change occurs in a region of the DNA that is responsible for proper brain function, you might get Alzheimer's disease or Parkinson's disease. So depending on where this mutation is present, you get different diseases. So you need to first map or understand how these genetic variation are linked to certain diseases, right? So there are two approaches people use to link the genetic differences in humans to a disease, right? So the first one is called genome-wide association study. It is done for very common diseases. For example, there are common diseases means that you see more commonly in the population that are such as type 2 diabetes, cardiovascular diseases, or asthma, Parkinson's, Alzheimer's, etc. And there is another approach used for rare monogenic diseases. So the rare disorders they're not very common. You see maybe one person in entire city has won that disease, and there are certain diseases that you may not know, but there exist a lot of rare diseases. And for example, muscular dystrophy, hemophilia, these are not very common, and they are very you rarely see them. And in this sort of diseases, they do something called family studies or trios studies or genetic linkage. So you identify all the family members and you sequence their genome and try to understand why only this family is getting this disease, but the rest of the people in the city are normal, right? So this is more focused, and this is a population scale level, and this is more focused studies. So I will just explain a little bit about what is this genome-wide association study. Basically, if you have people that have certain disease, and if you take people that don't have the disease, right? So you take 100,000 people that have type 2 diabetes, and 100,000 people are the same age but they don't have diabetes. Let's say you take 100,000 people of our grandmother's age, and you take 100,000 people of our grandmother's age that don't have diabetes, and if you compare their genetic sequences between these people have diabetes or people don't have diabetes, then you observe that there is something common among the people who have diabetes and that is missing from here. For example, here in this genetic sequence, you have G. So these are nucleotides, DNA sequences. You have G, more commonly seen here. But here you see more T. That means then you can hypothesize that having a G here in your DNA sequence might be responsible for developing diabetes. So that's how the genome association studies are done, taking thousands of individuals, trying to identify control people that don't have the same disease, and then do their genome sequencing or other approaches, and then compare their genetic sequences. Then you can see what are the specific nucleotide or genetic variation that are present in disease individuals, right? So the output of these studies is basically shown as this plot. It's called Manhattan plot. Basically, what you have here, you have 22 pairs of chromosomes, and each dot in this plot tells you what is the score of having a disease if that person has a specific genetic variation, right? So that's how these plots are read. This is pretty common plot that you see or you might see in future when you start looking at genetic data. But basically, this plot is telling at each point of your genome sequence what is the score. So this is represented as score of getting or having a diabetes. That means in the chromosome 16, there is a region in the DNA that has a high score, and if the people have mutations in that region, there is high chances of that people becoming diabetes patients, or most likely cause of getting diabetes is the changes in chromosome 16, this position in the DNA. So that's how these plots are read. So one classical example of how these genetic studies help us to identify drug targets. When people did, so this study was published in 2008 from Katsaren. He's in MIT Broad Institute in USA. So when they look at the cholesterol levels, so what they did is they identified people with high cholesterol and low cholesterol. So they identified 100,000 people that have very high cholesterol in their blood, 100,000 people that don't have cholesterol in blood. And then they checked what is the difference between the DNA sequences of people who has high cholesterol, who have low cholesterol. And what they identified is they identified several sequences or the regions in the genome, which is 30 regions in the genome that are linked or that seems to have high contribution or high score for having high cholesterol. So how to understand these plots is that when you have this signal, that means you go back to the genome and see where is this peak of risk, where risk is there, and you see what gene is there in the genome. That means if you have a peak here in the genome that has high cholesterol, you try to see which genes this genetic variation is impacting. So one classical example of this is that in this study, they identified this HMG co-receptor. There is a peak here. That means there is a high indication of the genetic differences in this gene causes high cholesterol. And a classical example of the drug discovery coming from genetic studies is that this HMG CR gene is already a target for statins. So people take statins to reduce their cholesterol levels, and these statins target this gene, which is also shown in the genome-wide association studies that there is a high signal in this gene that causes high cholesterol. So this is the first basic step of, or the first step of modern drug discovery, where you are trying to understand how the genetic variation is causing diseases and what genes or what proteins might be responsible for this disease. And then you do clinical trials, high throughput experiments, and like you follow all the protocol and you develop drugs. So this is another classical example of this genome association studies. So when they looked at the people with Crohn's disease, it's an immune disease, they identified there is a huge signal in the gene that is encodes this IL-2 receptor. So it basically, if you know about immunology, there are cytokines and there are different types of immune cells and how they react, and whether if they overreact, they do autoimmunity and they start destructing your own cell types or own tissues. So in this study, they did the Crohn's disease when they did GWAS. So they took 50,000 people with Crohn's disease, 50,000 people without Crohn's disease, and they look at their genomic sequence of their DNA. And they identified there is a huge signal in the IL-2 receptor, which encodes this receptor on immune cells. And this is very important for T cell differentiation. Again, it's I don't know how much immunology you know. So what they identified in this study is people who have less IL-2 receptor have less Crohn's disease. So then, obviously, this is telling very clear evidence that there is something to do with IL-2 receptor in Crohn's disease through genetic studies. And then later, there were already some drugs developed for this to target this IL-2 receptor for psoriasis. So what they did, okay, can we use the same monoclonal antibodies that is being used for something else? And they used these monoclonal antibodies to block this IL-2 receptor pathway, and that reduces Crohn's disease. So it's another classical example that the genes or the proteins that are highlighted by human genetic studies can be used as a starting point to develop highly successful drug candidates. So the previous talk we just heard, there is a classical way, there is modern way. Classical way is more heuristic and more random. And this is a very, very recent modern way of developing drugs, looking at human genetics data, identifying the most important candidates that show very strong signal. Then you target or try to develop drugs only for those things instead of like doing all random clinical trials, all random trials on all genes. So this is like cardiovascular disease, this is Crohn's disease. I just showed two examples, but this sort of studies have been done in more than 1,000 diseases. So people have done a massive amount of genetic studies in last 10 years that they have now identified plausible or more likely protein or gene that is implicated in any disease, and this is done for almost maybe 1,000 diseases. So we have a lot more genetic data now that has some suggestions of what genes might be responsible for what diseases. Now, what the next step is remaining is we need to look at these results and do some initial experiments to see how many of them are real, how many of them are spurious, then develop them for the drug, take them through the drug discovery process. So not drug discovery, but drug development process. So this is basically the first step of drug discovery. So why now pharma companies are investing a lot in genetic studies instead of doing any classical approaches? Because there was a paper published in Nature Genetics in 2015. This is paper from GSK, that when you look at all the drugs that are approved in clinic and ask how many of them have a genetic support or how many of them are randomly chosen, they observed that this is a score, and there is a highly likely that if a drug has a genetic support from the beginning, from these genetic studies, and so for example IL-2 receptor, when they do Crohn's disease GWAS, they identified IL-2 receptor as could be the important gene, and that has a high chance of being successful in the entire clinical trial and to the market. So this paper systematically looked at all the approved drugs and asked how many of them have genetic evidence or how many of them are supported by genetic evidence. And there is high chances that if the drug targets have genetic evidence, they are highly likely to be successful up to the end of the clinical trial process, not just phase 2 or phase 3, but they have chances to be in the market. So this paper is one of the key papers. Now the pharma companies realize that, okay, we don't need to do any classical, like the classical approaches. Now we are going to shift our focus entirely on human genetic data and try to dig or look for drug targets for different diseases. So now, in the last 5 years, all major pharma companies like Regeneron, Genentech, GSK, Pfizer invested billions of dollars in human genetic studies because that's where it's a gold mine to start with very highly likely successful candidates. So they're investing a lot in this human genetic space for drug discovery. But there is a lot of challenges, so it's very complex to understand if you don't have a background of human genetics. But basically, when you do these human genetic studies, the genetic variation that you identify are always not points to a gene. Sometimes they are somewhere in the genome that you don't know what they are doing, so you need to understand the signal and then understand which gene is actually acting through. And then there is a linkage disequilibrium that comes into play in the human genetic studies. And also, most of the signals are not strong. I'll just go back one slide here. For example, this is IL-23 receptor has very strong signal, and this is very strong signal. But you see, there is a lot of signals that are still important for the disease, but they're not very strong. So here you need to understand if it is a weak signal, is it a weak signal, or is it, or like it's complicated to understand what these weak signals are doing, but they're still important because they are shown in the studies. So so heritability is basically means how much disease can be explained is widespread across the genome. That means they have very small effects, and there are a lot of them, that it's very hard to digest all the information from all the diseases. And the gene regulation is too complex. Just I said that how does each cell know what it has to do, why pancreas secrete insulin, and why brain cells secrete dopaminergic neurons, and how it is controlled is too complicated. And it's very complicated, and there is different time points. It gets complicated. So to digest all this information coming from, even though human genetics is pointing out, already giving us a good candidates to start with, but it's still too complex to understand all the biology happening at the DNA level. So that's why now we are trying to do these large-scale, high-throughput experiments to validate the human genetic insights, then look for how many of these candidates are druggable, how many of them can be targeted by small molecules or any chemicals, how many of them survive in phase 2, phase 3, phase 4 clinical trials. So it's a process, right? So we are at the beginning of using this massive amount of human genetics data for drug discovery purpose, and all pharma companies are now really focused on starting with the human genetic data. They're not going to do any random experiments from now. They'll do experiments only if there is really strong evidence from human genetic studies. So maybe I hope this gave you a bit of brief overview of what people are trying to do, because if I go even more details, that is more complicated for you to understand. But I will give you 4 other examples how these advancements in human genetics and human genomics is trying to help us to have better healthcare. So the first case is gene therapy. So in gene therapy, what gene therapy means is there are some children born with a specific mutation. So when there is specific mutation, they don't produce specific protein. So how do you treat those children? Basically, then the gene therapy comes to rescue. So when the children has a specific mutation, for example, this is spinal muscular atrophy caused by a mutation in SMN1 gene. So if there's a mutation in the SMN1 gene, these children will not have a proper movement. They have movement disorders, and they don't live longer. So they die very early. So now when you do the genetic sequence of this child, when you take the DNA or blood or saliva and you put in the genome sequencer, you get the DNA sequence of this child, and they identify there's a mutation in this gene. Okay, so, and this child won't live longer if the mutation is there. So what scientists do is they take this gene or the protein outside, and they introduce into the child's cells, right? So this is called gene therapy. So they take the mRNA outside, so they synthesize the mRNA, they put into a vector, it could be a viral vector or some other vector, and then they introduce back into the child body. So they are artificially introducing proteins from outside, or not proteins, they are introducing the genes, because the proteins will degrade and there's a lot of complexity there. So they are introducing genes that are lacking in children's body. So this particular child, it was a news in the BBC a couple of months ago that this child was given this SMN gene therapy, and now he's active and he will live longer now. So this is one of the amazing examples of how understanding human genomics, human genetics, how the cell function at DNA level will help us to cure certain diseases that are not curable. If you go by classical drug discovery process for this gene, there are a lot of children born now who will die in a span of months or years, and drug discovery takes 20 years of time. So by using the human genetic or genomics knowledge, people are trying to come up with novel approaches to treat some certain conditions very fast. By the way, this cost like really, really expensive. The cost of this drug is almost 1.6 crores, no, 16 crores or something. So it's very, very expensive, and only I think Novo Nordisk or some companies producing these have this gene therapy drugs in the market. So the other example is precision medicine. So in precision medicine is what is basically meant is, so far what we do is, if there's a cancer patient, we give some medication to the cancer patient. If let's say the cancer is colon cancer, the cancer patient, if a guy has a colon cancer, we give some treatment. But the problem with giving the same treatment for all the same cancer patients is that this drug might be effective in some individuals, or this drug might not work in other individuals. Again, because the cancer is, again, mutations in the DNA, and depending on where the mutation is happening, some people, the same treatment might work for some people, or it might not work for some people. So you really need to understand what mutations this cancer patient has, and depending on the mutations and what gene is affected, what protein is really affected, you need to change the treatment. So now in abroad, it's very routine that when people have cancer, first they screen for the genetic mutations. So they take the blood or saliva, and they do the genome sequencing for the cancer people, and they look in the computer to see which genes are really affecting in different individuals, and then based on the genetic mutations, they choose different treatment options for the same cancer. Even though all of them has colon cancer, they give different drugs that might act through different pathways, because even though all of them have colon cancer, because different genes are mutated in different individuals, so you need different drugs. And this is done not only for cancer, but also it is done for other diseases. Like I think there is one classical example was warfarin. So the blood thinning agent warfarin also doesn't work the same way to all the patients. So when people undergo surgery, they take these blood thinning or thickening agents, and some of them also depends on the type of genetic background. The same drug might not work for everybody. So it's very important. It's the most important in cancer patients, because the cancer is all about mutations. So you need to really know where the mutation is, which genes are affected, and you need to choose a treatment depending on the mutations. Otherwise, the same treatment might not work. It might work in some people, but might not work in other people because of the genetic differences. So the other example is genome editing. So the genome editing, I think maybe, I don't know if you've heard, in the 2020 Nobel Prize was given for the technology, or the technology that is developed around genome editing. In the genome editing, what you can do is you can change the DNA sequence of a live person in a cell. So they have something called guide RNA. So it's again, the terminology may not be very understandable for you, but basically they take something called guide RNAs that cuts the genome or the DNA and introduces the correction. So if there is a mistake, you introduce something called guide RNA that goes to the DNA and it cuts it there. And because of the homology recombination or repair mechanisms, when the DNA sequence is restored, the error is gone. So you are correcting a sequence of genome, right? So that means if there is a defect because of these mutations, now because using this CRISPR-Cas9 genome editing technologies, you're correcting the DNA changes. So that has a lot of implications. For example, if there's a mutation that causes cancer, so you can do a targeted therapy of this CRISPR-Cas9 machinery to the cancer cells and correct the mutation in the cancer cells that will reduce the rate of expansion or rate of proliferation of the cancer cells. So this is one application of genome editing, but there is a lot of ethical concerns, because now what people are trying to do is, even before the baby is born, when the baby is in the inside womb, or when it's a very small embryo level, and if you do a genome sequencing of the baby, maybe there are some errors that might lead to diabetes of the baby in the future. So can you correct the DNA even baby is born, so that the baby is born perfectly healthy without any diseases? So that leads to a lot of ethical concerns. So how can you go around the ethical concerns and don't do something wrong that leads to like catastrophic defects or massive? So it's very dangerous to play around with the DNA sequence, because DNA sequence is the one that instructs everything in your life. So there are a lot of ethical concerns, but people are trying to formulate these ethical manuals to see where it can be applied, where it cannot be applied, whether it is really necessary to go to CRISPR-Cas9 genome editing, or are there any other alternative approaches to treat the condition. And if it is really necessary and it's very critical that there is no other alternative option, maybe they might think. But this is this is 2020 Nobel Prize, so we are in 2021, so it's very early stage to know how this technology is going to be used for curing certain diseases or certain conditions. And we have this genetic risk score. So genetic risk score is basically, because the information we already have so far from human genetic studies, we know which genetic changes or which errors in your DNA sequence cause which diseases. Can you predict what diseases you might get in future? Because if you take type 2 diabetes or cardiovascular diseases, you get them after the age of 45 years or 50 years, right? Cardiovascular or type 2 diabetes. But your DNA is there since you're born. So your DNA is always the same. Before 45 years or when you're 10 years old or 60 years old, the DNA is the same. And you develop certain diseases after a certain age of 45 years or 50 years. There are some Alzheimer's or Parkinson's, type 2 diabetes, cardiovascular. All these diseases show up when you're becoming older. So when you're younger, when you're born or when you're 20 years old, can we look at your DNA and assess how much risk you have for certain disease? Because we know what is the importance of some genetic variation for these diseases. Can we use that prior knowledge to inform you, okay, when you are 20 years old, I'm going to look at your DNA and say, oh, you have a risk of getting 37% risk of getting diabetes when you grow up older, or you have 90% chances that you might get diabetes because your genetics is telling me you are at very high risk. So that can be used. If you know that you have high risk for type 2 diabetes when you are 20 years old, you should be really careful, control your diet, control your carbohydrates, and do exercise. It's the same for cardiovascular diseases. If you have a high risk for getting a cardiovascular disease from after when you become 50 years old, and you know that when you're 20 years old, you can be careful, right? You can go for workouts, walking, exercise, playing like some sports, so that you become hot and you try to reduce the risk of getting cardiovascular diseases when you are 60. So this is very powerful information that we have, and it's very powerful way of knowing what kind of disease risks we have before even getting them, so that you can be a bit careful and try to reduce the risk when the time comes. So yeah, the genetic risk scores, I forgot something here. Yeah, so that's basically the genetic risk score. So you can estimate what is the risk for getting certain diseases right away, without waiting to know whether you get the disease or not. So that is another application of human genetics. So with this, I would like to finish, and I have like one last slide. So I have presented a general overview of human genomics, human genetics, and how it's related to disease. But we also did, in my PhD, I did a lot of work trying to understand genetics of type 2 diabetes, and we published several papers in like Nature Genetics, Nature Communication, Scientific Reports, Genes and Development. All these papers are slightly advanced our understanding of human genetics in relation to type 2 diabetes, because my my PhD boss is a world-renowned type 2 diabetes geneticist, and we did CRISPR-based genome editing studies in human pancreatic islet cells. We did a lot of genetic studies in large cohorts of people, and also something to do with the regulated genomics and etc. So this is my institute where I did my PhD, and this is my team, and these are all my collaborators and funding agencies that I worked during my PhD. And this is my Gmail, and this is my Twitter account. If you have any questions, if you want to know anything or any guidance to get into this human genetics or human genomics, because it's not really taught well, because it's being picked up by now in abroad. Like last 5 years, there is a huge amount of genetic data that is being generated in, especially in European and USA and UK. But in India, we are a bit slightly behind the we are slightly behind the technology, because it's very cost effective. It needs a lot of money. It's not easy to sequence 100,000 people. It will cost like few hundred, like 10 crores or 20 crore rupees. And so as we don't have so much funding and the technologies are so expensive, we are slightly behind these technologies. But if you want to know anything related to human genetics or human genomics, how it is being used, whether there are uses of studying it, so you can definitely like contact me through my email or my Twitter, or you'll find me. Just if you type my name in Google, you'll find me easily. So with this, I would like to stop presenting, and if you have any questions, I think we can take the questions right now or later. You can just ping me on my Gmail or Twitter. Thanks.
Yeah, thank you, Dr. Gautam Atla. Yeah, we have a few general questions. So one of the first question is, how the mutations are brought about so rapidly in viruses, especially in the COVID-19?
Yeah, so basically because the lifespan of virus is smaller, right? So when a person is born, like a human is born, the changes in the the DNA changes comes from father or from mother. So when the genome of mother and father combines, there are recombinations and everything. So there are mutations in humans, but the virus divides very rapidly, right? So the lifespan of virus is very rapid. So when it divides rapidly, every time the virus divides, so that means it is replicating its DNA into a new cell or new virus. That time the mutations are introduced. And why it becomes stronger? Because of the survival of the fitness. Because of the survival of the fitness, you always want to, when the virus is dividing, the changes that help the virus to live longer or live stronger will remain. So, and the weaker ones dies. So virus divides rapidly. Every time it divides very rapidly, there is a changes in DNA, and the changes in DNA that helps virus to become strong stays, and the ones that, so that's how the changes are very rapid, because obviously virus lifetime is very fast, like they can divide very quickly. And if the DNA material, the RNA material between two different viruses combined, that gives rise to like different strains, what we see now as a Delta strain or Alpha strain. So yeah, it's all about replication. The virus replicates faster. Every time you replicate, there is a mutations in the DNA randomly introduced.
Yeah, another question is, what are the methods available to study the genetic aberrations?
So I can tell only from the perspective of human genetic sequences, because genetic aberrations can be also carried. I think the karyotyping is also genetic aberration to see if you have a 23 pairs of chromosomes or not. So I think that's called karyotyping. But to study DNA sequences, we have something called sequencing machines. So where you take some cells from your body, either from your saliva, blood, or even any skin, whatever cell that is alive. You put that, you extract the DNA and you put that in a sequencing machine, and the sequencing machine will run for maybe a couple of days or 2 days or 3 days, and it will give you entire sequence of your genome. Then you can take the genome and compare to the standard genome and see what changes you have compared to the standard genome, and whether those changes are important or not important. That's what this genetic risk score will tell you. So yeah. So basically, you have this something called sequencing machines. They are called next generation sequencing machines, and they will tell you, they can sequence any types of material. For example, even they can sequence the COVID mRNA sequence. Right? That's how we discovered the vaccine, or that's how we say there's a new strain. How do we know there's a new strain? Because they take they sample the virus from different parts of the world, and they sequence the genome, and they see whether the genome is similar to the first virus, or is it so different. And that's how they say, oh, this is a new strain. So yeah, basically you need to use sequencing machines. Now people are using sequencing machines to sequence the DNA.
Yes, sir. One more question.
Yeah, sure.
Sir, are there any anti-diabetic drugs which are specific for the particular type of people?
So that so the drugs based on genetics, there is no diabetic drugs prescribed based on the genetics, but based on, so it's there are two types, there are different types of diabetes, right? The diabetes caused by less insulin secretion, that means there is a problem in the beta cell function itself. So whether you target the drug for more insulin secretion, that class of drugs. Then there are other class of drugs where you have normal insulin, but your muscle cells are resistant to insulin. So the insulin receptors are not so sensitive. So even though you have a lot of insulin, your muscle cells cannot utilize that insulin. So that's called insulin resistance. So in that case, you give a different types of drugs. Whether you are giving a drug for insulin secretion or insulin resistance, that depends. But prescription of drugs based on the genetic sequence is not there. I think it's not done routinely for type 2 diabetes, at least.
Yeah, it was an excellent presentation. Thank you for taking the time thank you for taking the time to speak us today on the very important topic of human genetics in drug discovery. Once again, I thank you, Doctor.
Thanks.
Yeah. Now let's move on to our final session. I would request Mr. Santosh, Assistant Professor, St. Peter's Institute of Pharmacosciences, to introduce our speaker, Dr. Raghavendra.
Good evening, everyone. It gives me great pleasure to introduce today's speaker, Dr. P. Raghavendra sir. He is here to talk about bioequivalence studies in drug development. Sir has completed bachelor's and master's from University College of Pharmaceutical Sciences, Kakatiya University, Warangal. Sir did PhD in neuropharmacology from University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Over 9 years of industrial experience in bioequivalence studies, bioanalysis of solid orals for different regulatory submissions like FDA, EU, WHO, ROW, Health Canada. Sir has published 7 international research publications and 2 conference reports. His research work was presented in various international conferences in India, Australia, France, and Argentina, and received travel grants from various international agencies. Sir has received Pratibha Scholarship Award from Andhra Pradesh State Government. Previously, he was associated with Milan, Hyderabad, and IPCA Laboratories, Mumbai. Currently, Dr. Raghavendra sir is working as lead researcher Strides Acrolab Private Limited, Bangalore. I request Dr. Raghavendra sir to continue the session.
Raghavendra, please unmute yourself. No, your voice is not audible. It seems there is a net issues. I think his device is not connected. No, your voice is not audible, Ravindra sir. So due to technical issues, we couldn't reach Dr. Ravindra. So let's move on to so his device is not connecting. So we come to the end of this webinar. I now request Dr. P. Rashekar, Principal LOC of this webinar, to give the vote of thanks. Dr. P. Rashekar. Now I am presenting the vote of thanks. At the outset, I would like to thank our beloved chairman, Sri T. Jayapal Reddy sir, for giving us the opportunity to conduct this webinar on such an important topic. We would like to thank our speakers, Dr. Kumar V.S. Nemmani, Director, Sri Vishnu College of Pharmacy, Dr. Gautam Atla from GSK UK, for giving us such wonderful lectures and enlightening us on drug discovery and development. We would also like to thank all the registered delegates from across the globe for participating in this event. As an organizing convener, it's my honor to thank Dr. P. Rashekar, Principal, St. Peter Institute of Pharmaceutical Sciences. We would also like to thank all the teaching and non-teaching faculty of St. Peter's Institute of Pharmaceutical Sciences for their support. Thank you one and all. Thank you everyone. We appreciate you being here. We hope to see you again next time.